Okay, so the purpose of this tutorial is to review the dihybrid cross from a chromosomal perspective. So we want to make sure that we're thinking about sexual reproduction in a biological way, and sexual reproduction starts with first you have meiosis, which makes gametes, then you have fertilization, which makes zygotes. So when we think about the dihybrid cross, we're starting with an individual that has some kind of genotype. Just going to use a general pattern, big A, big B, little b, and then we're mating it to another individual with that same pattern of genotype. So this is the particular cross that we would call the dihybrid cross. But what we want to think of when we do a dihybrid cross, or any cross really, is first think about what meiosis would make separately for each individual. So each individual will make gametes, and then maybe some of those gametes will get fertilized. But we've got to do first things first. So let's think about meiosis for each of these individuals. When you have an individual that's big A, big B, little b, what it means is in their cells, they have, let's say, a pair of homologous chromosomes, and let's say the gene A is on that chromosome, and they have a big A on one of their big chromosomes and a little a on the other one. And let's say for the Bs, let's put the Bs on a shorter chromosome. Here's a homologous pair, and you have a big B here and a little b here. So that's what the cells look like in terms of that chromosome layout. And of course, prior to meiosis, the chromosomes will replicate. So then you have the dyads. The sister chromatids are identical. And then during meiosis, each product cell has to get one long chromosome and one short. But when meiosis is going on for, many, many rounds, you can get gametes that have all different combinations that include one long and one short chromosome. So what we're going to say is that if we started with a diploid genotype, big A, big B, little b, then meiosis can produce gametes that contain a cell that has a big A on the big chromosome and a big B on the small chromosome. But it's also possible to get a gamete that has a big A on the big chromosome, but a little b on the shorter chromosome. And it's also possible to get a cell that gets a little a on the big chromosome and a big B on the little chromosome. And it's also possible, and I'm sorry, I'm running out of space, to get a little a and a little b. So I'm not saying that one meiosis would generate those four gamete types. I'm saying that after thousands of meioses, you would have approximately equal amounts of these gamete types. And so instead of drawing this all out, what we usually do is just write the letters. So some gametes will have big A with big B, and I put a comma. Some gametes will have big A with little b, comma, little a with big B, comma, little a with little b. Since the other individual that's involved in this mating is the same genotype, then it would be the same four gamete types. So these are your gamete genotypes. And what we indicate here, we're just using the letters, but what we have to be thinking is that the A marks the long chromosome and the B marks the short chromosome. So every gamete's genotype includes one long chromosome and one short. So each gamete has a haploid genotype, whereas each somatic cell has a diploid genotype, two copies of each type of chromosome, two longs and two shorts. Now, when a gamete that has a big A with a big B is able to then meet or fertilize with another gamete that has a big A and a big B, then when those two come together, we end up with a zygote, because fertilization makes zygotes. And the zygote has a big A, big B genotype. And we continue through, let me see if I can erase all this. I'm going to do it this way. So when we, once we figure out our gamete types, all we have to do is put them on the Punnett square grid to figure out what our zygote types are. So that was one way to work it. So the Punnett square is just a way to keep track. If we have some gametes that are big A, big B, some gametes that are big A, little b, some that are little a with a big B, and some that are little a with a little b. And we put the same thing down here. And if you want to fill this all in, inside the grid, fill in all the the zygote genotypes, which would be inside the grid, you are most welcome to. But what I can assure you is that out of these 16 squares, 16 zygote types, out of 16, 9 of them will end up with a genotype pattern with at least one big A allele and maybe another big A or a little a, and one with a big B allele, and maybe a big B or a little b there. 3 out of the 16 will have a pattern that has a big A and either a big or little a, and a little b, little b. 3 out of 16 will be little a, big B, and then either big or little b. And only 1 out of 16 will be all lowercase, all recessive. So if you're given a cross that fits the dihybrid pattern, then you can solve it this way. This is the long way to go. But if you recognize that pattern, then you can just skip to the punchline, which is that the offspring would fit into these patterns. So 9 out of 16 would have a dominant phenotype for both traits. 3 out of 16 would be dominant for the first trait, recessive in phenotype for the second. 3 out of 16 would have a recessive phenotype for the first trait, but dominant for the second. And 1 out of 16 would be recessive for both. So it's much easier to recognize the patterns and learn the dihybrid cross this way. This is, for any 2-gene cross, this is the most complicated way it can go. Right? But it is called the dihybrid cross, and it is one of Mendel's.